Hello, my name is Christopher Bravery. I'm a consulting regulatory scientist at Advbiols. And this is a bite-sized regulatory series where I hope to provide a series of short presentations on the common technical document. This first one intends to orientate those who are not familiar with the common technical document to help them understand the overall structure. In subsequent presentations, I will take subsections and try and explain the sorts of data that should be in them for cell and gene therapy products. This could be useful to people in biotech, but it's very much aimed at the cell and gene therapy community. So hopefully you can see the screen and you can see the web link, which is advancedbiols.com, and then to the resources tab. And then if you go down, you'll see there's a section, there's a link to something called the common technical document, and it'll give you a version number. This is from around about a year ago. I haven't had a chance to update it any further, but hopefully it's already useful. And this is the broad structure of the common technical document. And if I open this here, in fact, I've included the other sections just in overview for those of you that aren't familiar. So the common technical document comes from ICH, which is an attempt to internationally standardize a technical document that can be used with all regulatory authorities. And we've now reached a point where most of the regulatory authorities, the major regulatory authorities, are using it. Certainly the FDA, Japan, EU, etc., are all using this document. Australia, New Zealand, and so on. It's generally used by ICH regions. So in principle, you can use the same dossier in all regions, although there's always some details that differ from country to country. But that is the intent, is to harmonize it. Now, Module 1 of the Common Technical Document isn't technically part of the Common Technical Document. It is the administrative information, which is very much country-specific, the various forms they require, etc., for your submission. Module 2 are a series of summaries. So I certainly won't be discussing those to any degree. And then we have Module 3, which is quality. This has historically, with the FDA, have historically called this section chemistry, manufacturing and controls. And that term comes from their original standard dossier, which has now been superseded by the Common Technical Document. So I'm sure we'll still continue to call things CMC, but actually now under the Common Technical Document, it's called quality, which is actually the term that's generally used in Europe. Module 4 is the non-clinical studies. So that's both the preclinical animal studies and other in vitro studies, but also those that may have occurred after clinical development has started. So that's primarily around the safety, but also the mechanism of action of the product. Module 5 is all the clinical studies. And I haven't prepared a mind map of this nature or for these sections, because generally those section headings are much more straightforward. But the module 3 that describes the manufacturing aspects is somewhat more complicated. And in particular, if you're not familiar with it, it's worthy of a quick explanation. So aside from the fact that there are a sort of a regional section for those things that weren't agreed internationally that some regions may want. And then there's a literature section, because you should provide copies of all your literature that you cite with your dossier in the dossier. Actually for clinical trials as well, really, but certainly for market authorization. There's a table of contents. But 3.2 is the main body of the data. And it is divided into 3 sections. 3.2s describes the manufacturing, control, and quality of the drug substance. So that's the active substance. In GMP guidances, this is called an active pharmaceutical ingredient. And sometimes it's called active ingredient. So it has multiple names, but in the ICH dossier, it's called the drug substance. The drug product is the final product that you release. And this may contain one or more active substances. Most of you will probably only be familiar with it containing one active substance, as indeed am I. I can't particularly recall working on a biological product that had more than one active substance, or not deliberately so. Possibly, but not for not in a deliberate sense. And then the appendixes I'll come to briefly to explain in more detail. So if we just sort of position that at the top and then open the drug substance and drug products sections. Now, wherever you see a little paperclip on my mind maps, that will link you to another mind map, which gives you further information. Whenever you see, which I can't immediately see now, if you see this little note symbol, if you click that, the note will come on to the left, but I shall try and avoid doing that to confuse you too much during this presentation. So the first thing you do is, and I've deliberately tried to align some of the coloring to make it a bit easier to see it, but maybe if we go in reverse order, you'll see both sections have a stability section because you need to know the stability of the active substance, which is made separately from the drug product, and the drug product, which will be a different concentration and in a different and in excipients, whereas the drug substance is in some sort of buffer or whatever you like to call it, a different formulation. Both sections, the material needs a container closure, so they both have a container closure section. Then there's reference materials for the analytics of both sections. And then there's a section on the control of those substances, either drug product or drug substance. Skipping up, there's a section for manufacturing of both of them. And then at the top, there's a very simple introductory section which just provides some high-level information. And they're fairly similar. So if I just sort of open up what they ask for in S1 for the active substance nomenclature, so what do you call it? What other names has it got? What's its description, etc. The structure, well, that's tricky. It's a little bit trickier for cell and gene therapy, but obviously if it was a small molecule, you would just draw a chemical structure. And if it was a biological molecule, you'd provide the amino acid sequence and so on. And then the general properties. And again, this can be a little bit difficult for cell and gene therapy products to decide exactly what to say, but it would be things, some sort of maybe the phenotype of the cell and that sort of thing. So it's just a little bit of information to orient the reviewer or assessor. And there's similar headings that you would expect in P1, which for some reason I haven't actually included. So let's look at manufacture. And I suppose I should just demonstrate to you that the manufacturing section in the P section and the S section are quite similar, but with a slight difference again. And I'll try and explain why that is. And I apologize for all that stuff, but that has a relevance too. So the first heading is the manufacturers. There's usually more than one organization involved in manufacturing, so you have the same there. Then a description of the manufacturing process. So again, you've got a parallel. Control of the materials, you don't have one of those in P because your materials are excipients and they have a separate section. And then the control of critical steps and intermediates, again, that has parallels. Process validation has parallels. And then there's a manufacturing development section here, which is in a different place in the drug product. Not quite sure why, but obviously, obviously when something's put together by committee, sometimes things are not quite as rational as it might seem to somebody from the outside. But it's not that difficult to get your head around. So this is all just really about explaining who manufactures, how they make it, how they control the materials they use, how they control the process, how they've demonstrated that control of the process through validation, and then some history of that development. And likewise here, except the history of the development is tucked up in P2, which I'll come back to in a bit. Then the characterization here is really characterizing the active substance and the drug product. The only difference potentially is the drug product might have some different impurities introduced through the drug product manufacturing. So, and I should just re-emphasize, if you're developing a cell product, you won't necessarily be able to define a drug substance in a stable form that can be held. In fact, I would almost certainly recommend that you don't, unless there's some good reason to do that. And you would just describe your manufacturing continuously through to the drug product. And in a different video, I will talk about that. I'm not going to get too bogged down with that now. The control of the drug substance is mirrored with the control of the drug product. And again, the subsections are pretty much the same. The first thing you have is the specification for the release and actually for the stability of the product. That should be in that section. So either S41 or P51. The analytical procedures used to do those release tests. The validation of the analytical methods used to do those release tests. A list of all the batches manufactured and the release results you got. That's actually a very important section. And even in a clinical trial application, there should at least be some data there, because the assessor needs, or the reviewer needs, to understand how consistent your batches have been in quality. And then the final section is to justify the acceptance criteria and actually the methods that you've used in order to release the product. So ultimately, this is really where you justify that your quality attributes that you're using are actually critical quality attributes, and that the ranges that are allowable ensure that the product is of an appropriate quality and assures safety and efficacy. I won't talk about reference materials, container closures. It's fairly obvious, and I'll talk about those in detail in subsequent sections. Then both the drug product and the drug substance section obviously have a stability section. It starts off with a detailed conclusion of what you think the stability of the product is, your expected shelf life, your in-use stability, your accelerated degradation, your and all that. The middle section, the post-approval stability commitment, is not applicable in clinical trials and is something that you negotiate for the market authorization, how you will address stability moving forward. And then the third section is just actually like the batch the batch data section. It's really just the tables of results so that the assessor or reviewer can look at it. Characterization has no particularly, has only two sections defined. The thing to remember with the Common Technical Document is at the level in which it's described by ICH, so in S3 to the level of S3.2. But beneath 3.1 and 3.2, you can create whatever subdivisions you like, as many as you like, in the logical format that makes sense to you. Characterization is divided into elucidation of structure and other characteristics. And I think for biological products, I think that's better described as elucidation of structure and function. For impurity, and then the other section talks about impurities, and that's all the types of impurities. And they're the impurities of the active substance. So the thing that's particular, there's a couple of things that are different about the drug product section. The first one is P2, which is pharmaceutical development. So overall, P2 is a little bit like S26 here, manufacturing process development. The whole section really is a bit like that. So it starts off with a relatively simple sections just that don't have much detail in them. They're just asking what are the components of the product. And then you go into the drug product itself, and you've got a section on formulation development. Oops, well, I won't drown you with that at the moment. So formulation development is explaining the studies you did to identify a formulation that's both safe to administer to humans and also gives you the best stability of your drug product. And then a few other details that it's not really worth dwelling on at this point. And then your manufacturing process development here is the one that's directly comparable to the manufacturing process development of the active substance. Some other stuff about the container closure. In the subsequent videos, I'll explain how that differs from P7. The microbial attributes, not really applicable to biologicals, and the compatibility of it with the container that you're using, etc. And then the other key difference, obviously, is that you don't have a materials section like the S23 of the S section. But what you have, of course, are excipients, because this is now the drug product. So the definition of an excipient is anything other than the active substance and any devices that you might have as well, and the final container. Everything else is basically considered an excipient, although I will warn you that there could be variations in the interpretation of that regionally. But the ICH definition would be that. So this is really, so the specification are the specifications of all the individual excipients. Then where you do some in-house testing of those excipients, which you should at least have an identity test, and you'll probably need to do more tests than that by approval. Those analytical methods would be here, plus their validation. Then justification of the specifications of those excipients, because they're going to be administered directly into humans, so that justification is somewhat different than it would be for a raw material. And then some other stuff about specifically about risky excipients, if you like. And that's it. So they are quite analogous. It can seem baffling at first, but hopefully that gives you at least a hint that it's not quite that complicated. The appendixes include information on the facilities and equipment. At the moment, I don't have much detail in there, but I may address that in due course. Then there's a very important section, A2, Annex 2, which is about the adventitious agent safety, and which is critical, of course, for biological products, which include cell and gene therapy. And that's where, if you have any viral clearance steps, etc., they're described, and then you describe the adventitious agent risks of the various materials you use, both excipients and raw materials. And then that separate section for excipients is actually more about novel excipients, etc. And that's it. So I think that's a good place to stop. And I hope that gave you a little bit of a flavor and a very quick walk around the CTD to give you a little bit of a feel for what those sections are meant to be about. And do look out for more detailed subsections. Thank you very much.